they would slowly improve the human species. The genome is the complete set of genes in a cell or organism. The eugenicists advocated selective breeding for humans, as has long been done with farm animals. To influence human reproduction, various policies were put in place in the United States and Europe, such as forced sterilization of those perceived to be genetically inferior, restricting immigration from countries whose people had undesirable genetic qualities, and thus preventing potential mating and offspring, and generally encouraging better humans to have more babies through moral suasion and propaganda. There was very little understanding of what was actually a heritable trait and what was just a reflection of the social biases of genetic scientists. After all, this was an era when it was thought that some people's genes made them moral imbeciles. In this era and into the 1940s, what came to be called the mainline eugenics movement assumed that positive and negative traits were structured by race and class. This logic reached its height with the Nazis, who presumed that the Aryan race had superior genetic traits and others had negative genetic traits. This thinking provided the scientific justification for the Holocaust, when millions of people from, quote, genetically inferior, unquote, races were killed. This book is about the debate over genetically changing humans that started in the 1950s, after the discrediting of coercive race-based eugenics that reached its apotheosis with the Nazis. Scientists still had little idea of how genetic inheritance actually worked, but this changed with the 1953 discovery by Francis Crick and James Watson of the chemical structure of deoxyribonucleic acid, DNA. It turned out that the elementary units of inheritance, by then called genes, were just particular sequences of chemical bonds. This reinvigorated the chastened eugenicists, who still nurtured the dream of creating an improved species, albeit now through voluntary, racially neutral eugenics. Yet no longer would advocates have to develop various mating schemes to get the right people to have more babies. If the genome is merely chemicals, improved genes could just be inserted. Scientist Robert Sinsheimer wrote during this era that the new technologies allowed for a new eugenics. The old eugenics would have required a continual selection for breeding of the fit and a culling of the unfit. The new eugenics would permit in principle the conversion of all of the unfit to the highest genetic level. For we should have the potential to create new genes and new qualities yet undreamed in the human species. As one would expect, there was an ethical debate at the time about whether we as a society should engage in HGE. I will fill in much detail in the following pages, but for now I will jump forward to today. Knowledge of what particular genes do for the human body has increased dramatically, but the ability to correctly insert a gene into an individual's genome has remained elusive. For many years, scientists tried to heal people who had genetic diseases by getting a proper version of the gene in question delivered to the malfunctioning cells in their body. The delivery vehicle was a virus, which itself sometimes caused problems and, more importantly, often delivered the gene to the wrong place on the genome, causing another set of problems. To overgeneralize, from the 1960s to the 2010s, HGE would not work. Unable to even heal genetic diseases in people, Sinsheimer's dream of improving the human species remained fanciful. The debate about the ethics of HGE lost energy as it appeared that the most controversial applications of this technology would be impossible. I contributed, in a very small way, to the decline in the debate. My first book, published in 2002, was a sociological explanation of the ethical debate about HGE up to 1995. Shortly after publishing that book, I concluded that the more controversial applications of HGE would always be impossible, and I moved on in my career to related topics I thought more relevant. My decision was premature. In the early 2010s, scientists invented a new gene delivery system called CRISPR-Cas9. It removes the unwanted part of a gene and then places a new part where it is supposed to go on the genome. How CRISPR works is not important to this book. All you have to know is that it is already being used in humans. This precision is reflected in the desired metaphor of the scientists: human gene editing.